Thanks for the invitation to speak. It's a pleasure to speak for the EPFL. So Michael asked me to kind of give a broad introduction of what we do for people from imaging, various grounds of backgrounds in imaging. So to start with that, yeah, as you said, I work at the Laboratory of Molecular Biology, and the main remit of our lab as a whole is to understand the molecular processes that underlie life on Earth. So that the molecules that are responsible for much of life on Earth are biological macromolecules, and proteins are among the most abundant ones, together with ribonucleic acids and lipids, etc. And it's this complexes between these biological macromolecules that fulfill all the different tasks in human cells or other life form cells that make life possible. So I just thought, just to put things on a scale, we ourselves are at more or less at the meter scale. We have to go down in steps of 100 to our finger paper thickness, and then we're coming into unicellular life forms like bacteria, which are more or less on the order of a micron. So if you go another factor of 100 down, then you're about a 10 nanometer type of scale. That's the size of proteins, which themselves are made up of thousands, tens of thousands of individual atoms that are bonded together to form these molecules, and atoms themselves are in the order of an angstrom, or 1/10 of a nanometer. So your own cells then are multiple micrometers large. You can look at those in a light microscope, but if you want to go to smaller things like viruses, protein molecules, etc., then because of the wavelength of light being several hundreds of nanometers, you can no longer directly use light to look at the details of these molecules, and that's why we use electron microscopes. So this is a picture of our the first Titan Cryos electron microscope that was installed in our building when we moved back in 2013, and this is Xiao Chen, who's our EM facility manager, just next to it to kind of give you an impression of the scale. Now, many people in our institute, they purify protein molecules from many different organisms and in many different ways, but they get a little tube with a liquid in which these molecules, if you like, tumble around, many copies of the same molecule enriched from their original source. These are put on what we call an EM grid, which is then put in between tweezers. We put a little droplet, about 2 or 3 microliter on this grid, and then we use filter paper, and we have a robot to do this, to kind of blot away excess liquid, and then we very quickly dump this grid in liquid ethane, so quickly that the water that the protein molecules tumble around in doesn't have time to crystallize, and you get this amorphous state of solid water called vitreous water. Now, the grid is made up of holes. I'll have some pictures later, and the water kind of spans in between these holes and forms a layer of this now vitreous state of water, which is hopefully on the order of 1 protein molecule thick, so that you have a single layer of protein molecules suspended in this water, thin layer of frozen solid water. And this is then imaged inside the microscope. So I thought, just show you, this is a picture Chris Russo took of a grid. This one's made of gold, so you have all these grid bars. These are just little gold bars which make a rectangular grid. The thing is about 3 millimeter big, so you can just pick it up with a pair of tweezers. Now, if you zoom in, you can see that on top of the gold bars, there's deposited a carbon film in this case, and the carbon film has holes of about 1 or 2 microns in them. Those are these little circles. So if we now want to zoom in, we're now going to use electrons. This is an electron microscopy image. You can see the holes a bit closer. These ones are probably about 2 microns across in diameter. And we can now zoom in and take a picture of this thin layer of vitreous water in which protein molecules are now they were suspended and they were frozen by dumping it in liquid ethane. And to make this easier for you, I've put kind of black circles and you can see some of these molecules. You can see the amount of noise in the images is high, so it's a rather gray image, and the individual proteins are quite hard to see. They're black now. I'm going to invert the contrast in the next images, but you can see that it's not very easy to see where they are. And this is now an image of one of these individual molecules, and just to show you again the noise. The reason for that is that the electrons that we use to image directly damage the molecules. So you get, if the electrons hit the protein molecules, you get radiation damage. So bonds start to be broken, and then radicals start to appear, and they cause secondary havoc and so on. And in the end, the entire structure you were interested in imaging kind of disappears from radiation damage. So it's a bit like you yourself lying under the sun. If you want to avoid that, you shouldn't lie too long under the sun. So in the electron microscope, we only use very short exposures with a limited number of electrons to get these images. And then simple counting statistics, for example, together with the relative low contrast between the protein molecule and the surrounding water, result in these images where you have a large amount of noise. But because protein molecules in solution, you have many copies of what you hope is all the same molecule. And if you can determine images which are in the same view, because the molecules were tumbling around in random orientations, hopefully. So you'd have top views and side views and front views and all kind of intermediate views in those images, provided you can now determine which ones are top views and which ones are side views. And so you can align these, because you have to rotationally, translationally put them on top of each other. Then you can start doing averaging. So here I've inverted the contrast, so now the signal is white, and this is now the average of 2 of these images. And already you can start to see a little bit more signal. This is the average of 5 of these individual images, 10 of them, 50 of them, 100 of them, and 500 of them. And you can see that in the end, I'm getting rid of quite a lot of noise. Of course, that's stochastic, the noise. You have different instances in each image, whereas the signal hopefully is the same, so that survives the averaging. And you can now start to see all kind of internal structure within the overall shape of the protein. Now, because, this is 1,000, because the protein was tumbling around in many different orientations, provided you can separate all of these out, you can then calculate average for all of these, which is what you see here. So you have now views from something which is probably kind of an elongated molecule, but you also have some front views and side views, etc. And you can combine all of these into a 3-dimensional reconstruction. For that 3-dimensional reconstruction, the actual averaging in 2D isn't necessary. You can do this on the individual particles. How does that work? For that, it's good to know the type of images that we actually get in this form of imaging, which is transmission electron microscopy. So I have a 3-dimensional object here. Then if I'm imaging this with the electrons coming here from the left to the right, and I get a projection image on in the microscope. So this is the same as putting your hand in an X-ray generator in the hospital. That's the type of image that you get. But then instead of your bone density, you would get that type of projection image, an X-ray integral image, if you like, from this electron scattering potential that is caused by the atoms in the protein structure. So X-ray integral image, kind of an accumulation of all the scattering potential along an integral along the lines, is the 2D image. Now, the projection slice theorem says that this projection image in 2D, you can take the Fourier transform and you get a 2-dimensional Fourier transform of this 2-dimensional image. The original 3-dimensional object that this projection came from, you could have taken the 3-dimensional Fourier transform. Now, the projection slice theorem says that, that the 2-dimensional projection, the Fourier transform of the 2-dimensional image is a central slice through the 3-dimensional transform of the original 3D object. And the orientation of the slice is perpendicular to the projection of the electron projection direction. So that means if you know for all of your different 2-dimensional images how they are oriented in 3-dimensional space, if you know which one's a top view, which we know which one's a side view, front view, etc., then you can start to fill in the three-dimensional Fourier space just by adding up these two-dimensional Fourier transform slices in the three-dimensional space. You populate the entirety of 3D Fourier space, and then all what you need to do is an inverse transform to get to the three-dimensional reconstruction. So for the object that I just shown you, which was this kind of elongated protein molecule, this is a example, a three-dimensional reconstruction. This molecule is called dynactin, and it is involved in the transport of molecules throughout the cells. Now, the 3D density now here has been converted into a kind of a cartoon representation of an atomic model. So just to show you, this is then kind of a very small zoom up, and this is now 1/10 of a nanometer, or 1 angstrom, which structural biologists like to think of a type of scale. So this is a very small zoom in of that three-dimensional reconstruction, and structural biologists then build atomic models of the sequence of all the amino acids of the protein inside a three-dimensional map, and that's what we call the atomic structure of this molecule. Now, that then gives us an idea of the three-dimensional arrangement of all the chemical entities of this protein, and can help us understand how these molecules function in the living cell. For example, and this is not my own work, this is from Andrew Carter's lab here down the hall at the LMB. This dynactin molecule is depicted here in green, which is involved with coordinating contacts between this motor protein, dynein, in blue, and some connectors which are connected to cargo. There's a big balloon of cargo, if you like, and this dynein molecules pulls this balloon through the cell along these roads of the cell, which are called microtubules, and dynactin kind of ties the string of the balloon to the motor, if you like, and is thereby involved in transport throughout the cell. So these are the kind of things that we want to do at the Lab of Molecular Biology. And I would say in general, this is a goal of structural biology, to understand fundamental processes in the living cell at the molecular level. Okay, so this field of cryo-EM reconstruction underwent rapid progress around 2012, 2013. And this plot illustrates this from an old review that we wrote. All the black points were entries to the electron microscopy database of people submitting their structures in the years 2000 to 2010. And the larger, this is the weight of the protein, the larger a protein is, the better resolution in general you could get, because there is more signal compared to the amount of noise in the images. But all the resolutions that we got were in the range of 2 nanometer, 1 nanometer, perhaps a bit beyond, but more or less you would get a blob of density for protein domains, or perhaps if you were lucky, you'd get kind of tubular densities for certain features at a smaller level, which we call secondary structure of the proteins. But it wasn't until 2013 when suddenly these red dot structures became available. So suddenly we had reconstructions at about 3 angstroms resolution. And that is the type of detail that you need in a three-dimensional reconstruction to be able to build these atomic models, because then suddenly you see all the details that you know where individual residues should go, and then combining that with prior knowledge that we have about how amino acids the structures of amino acids are in proteins, you can then propose reliable atomic models from these 3D reconstructions. So what happened? So this was two main things happened. One was the availability of prototypes at that point in 2012 of a new generation of detectors. Until then, we'd been collecting mostly on photographic film, and the detective quantum efficiency, or if you like, a very shortcut way of thinking about it, the fraction of electrons that get detected correctly on the detector for photographic film was about 1 in 4 or 1 in 3, let's say. And suddenly, with the availability of new digital detectors that were specifically designed to detect electrons directly, that ratio went up to 1 out of 2 of the electrons, or perhaps even higher for certain types of detectors. And This was one development which was kind of pushed for many years by Richard Henderson at the LMB, who in 2017 won the Nobel Prize for his contributions to cryo-EM structure determination, together with Joachim Frank and Jacques Joubouchet, of course, who I don't need to introduce in Lausanne. So that was one thing that happened in 2012. There was the first prototypes of these Falcon directors, the K2, etc., became available, and suddenly we could do this. Now, these were different from photographic film, in which we had a single exposure, because just like your photo camera. But the problem is when electrons hit these thin samples of frozen water with proteins inside, that the radiation damage not only damages the protein, it also causes the whole sample to move around. And so what you're basically doing, you're taking pictures of moving objects, and you would have to take a 1 second exposure. If within that 1 second, your whole molecule moves by a nanometer, then that will, of course, limit the type of the resolution that you can get from the reconstruction from those images, because the whole motion will blur out the image. Now, the new detectors were digital cameras, not unlike the ones you have in your phone, which you can put in burst mode. So now suddenly we could collect movies, and the movie frames could be aligned to kind of correct for that motion, and that gave even higher resolution pictures. Another contribution to image processing was the development of the regularized likelihood approach in RELAM. And that development in RELAM kind of represents half of the work currently in my group. So I have about 4 people working with me together on algorithm development inside the software package. And then there's another, the other half of the group works together with people from Michel Gurrit groups in the neurobiology division on actually applying the methods that we develop in the image processing to a specific type of problem in structural biology, which is reconstruction of amyloid filaments. And I'll tell you more about both of those. So RELAM is our software package. It actually stands for regularized likelihood optimization, because that's what it does, as I will go over in a few minutes. We do this proudly open source, because we think that by keeping the software contributions to the field completely open to people, everybody can build on each other's successes, and the field will be able to continue to move forward as rapidly as it has done in the past 2 decades. So if you don't mind ranting me about how open software accelerates science, then you can follow me on Twitter. So the bad news about the reconstruction problem that I've just described you is, because of those high amounts of noise, and because we don't really know which ones are top views and side views, etc., we kind of need to align these images. That makes for an inverse problem where the experimental data alone is not enough to determine the unique solution. So in other words, there's many noisy reconstructions that describe the data that you've observed equally well. And that brings, if you don't treat that well, then you might end up with very noisy reconstructions. And then we humans are great at inventing things we hope to see in noisy patterns. So there's a danger of incorrect interpretation in those noisy maps. Now, there is good news too, because if you can incorporate external information that you have about cryo reconstructions in addition to the experimental data that you're about to record, then you can express the whole reconstruction slightly differently into a problem that can be solved, and that for which a unique solution does exist. And that in general, I guess, is called regularization. And conventionally in the field before, say, 2012, people would regularize their cryo-EM reconstructions in kind of ad hoc manners. People would put low pass filters on them, or they had all kind of ad hoc Wiener type filtering approaches. So what I set out to do in Cambridge was to kind of put then that reconstruction problem in inside a Bayesian framework. So Bayes' law here says the posterior is the likelihood times the prior divided by the evidence. Now, the evidence, the probability of observing the data perhaps depends on how much money you have nowadays, but once you have the data, it's kind of irrelevant. So we can optimize the probability that a certain model that we have is the correct one giving the data by optimizing a regularized likelihood function, the likelihood function being the probability of observing the data given the model, and the prior being the probability of that given model in that comes from sources that are not your data. So what do I know about Cryo-EM reconstructions in the absence of data or before I collect any data? So to do this, I formulated the problem in Fourier space because of that projection slice theorem that I spoke about earlier. Makes things very easy. It just becomes, basically reconstruction becomes a summation in Fourier space. So for that, I assume that the noise is Gaussian distributed and independent between different Fourier pixels, and that it has a certain power spectrum which can vary with the spatial frequency of the Fourier component, and that's expressed then in this formulation. The independence then allows you to multiply the probability of observing each individual pixel value by all the pixels in the image, and then it's just a Gaussian function where the experimental image, the Fourier pixel of the experimental image, is compared with the projection in a certain direction of the volume, and that's then multiplied by a contrast transfer function which describes the optical system of the microscope, and the rest is just a Gaussian. Now, because we do not know the orientations of the images, I do not know which is the correct orientation. Is it a top view? Is it a side view? And this could also include translations in the plane of the image. I need to kind of put the molecule in the center, if you like. So this phi parameter here could in 3D reconstruction would be 3 Euler angles, if you would like, and 2 in-plane translations. And the way we handle that is we just marginalize over them. So we calculate probabilities for all of them, and also there we then have some opportunity to inject prior information on what we know are likely orientations or unlikely ones. For example, one that is used is that translations, in-plane translations, are very likely to be Gaussian, well, we expect them to be Gaussian distributed, centered at the center of the image, and then very large translations are not so likely. Okay, now that's perhaps relatively easy to come up with a likelihood function. That's just what, if you know the kind of what happens, the image formation model in the microscope, and you have some idea of the noise, type of noise, then you can formulate your likelihood function. But of course, what do I know about reconstructions in the absence of any data? That's a much more difficult question. So ultimately, we know a lot about protein reconstructions. We know that that proteins are made of amino acids, and that amino acids adopt certain domains, protein domains, and they're made up of the secondary structure elements that I mentioned briefly earlier, alpha helices and beta strands, etc. Now, to express that mathematically is rather complicated, although we're now trying to work in that direction too. The original version of our program, which is still used to date, has a much less information-rich prior, but it's easier to work with mathematically. And that is, we assume that the Fourier components in the reconstruction are also Gaussian distributed and independent, and we actually limit the power. So by limiting the power of higher spatial frequencies in the reconstruction, we're basically imposing smoothness in real space. So all the information that goes in is kind of saying to the reconstruction program, do not make very noisy high spatial frequencies if the data does not support it. That just gets expressed in a Gaussian function like that. Now, if you combine the marginalized likelihood function with the prior, then you have your regularized likelihood target, and you then apply an optimization algorithm to that. And we've mostly used just straightforward expectation maximization. And then you get an update formula where you can calculate reconstructions from what people may already recognize as kind of a Wiener filter. But this Wiener filter, unlike conventional Wiener filter approaches that were used in the field so far, estimates the parameters of the filter directly from the data themselves. So we estimate how much the noises in the individual Fourier components of the individual experimental images by comparing the power of the difference between the images and their reference projections, if you like. And then we estimate the power of the signal from the reconstruction itself. This is prone, it's possible to overfit this, but then we have other tricks to kind of keep that at bay. And then these are combined into this optimal Wiener filter, which now does the best reconstruction under the assumptions made, and the parameters for it are estimated from the data. Okay, so until now I've only spoken about orientations in terms of the marginalization. Now that was all based on a comment that I made earlier on in saying that we hope that all the copies of the proteins that are on my experimental grid and that I've imaged are all copies of the exact same molecule. They may tumble around in different orientations, but the molecule itself is a rigid three-dimensional object that doesn't change. Now that is very much untrue for many biological data sets that we, for many biological objects that we image. And that is because protein molecules or biological macromolecule in general, they often adopt multiple different conformational states in the functions that they perform. And that is because many of these molecules, they really act a bit like the nanoscale equivalent of machines that you're familiar with in normal daily life. And this molecule is one example for that. This is an ATPase. And basically it works as a molecular equivalent of a water mill, if you like, where you have rotation along a stator and it's involved with energy generation. And so there's different rotationary states in this type of molecule. It can turn around in different states. And when you image these kind of molecules, very often you then get a mixture of all these states in the micrographs. But by now also marginalizing over class assignments, we can basically do classification. So the computer can find out which of the individual particles in these micrographs corresponds to each of the different states in the data. And you can then separate, you can classify the data, in this case, for example, in 3 distinct states, and solve structures for each of the 3 states from one data set. Okay, now one problem that many people in the field now are interested in is if this is not a discrete number of states that exist in the data, but it becomes a continuum. For example, a continuum rotation around this axis, or there could be some arm waving around, etc. And current methods, many of which are still focused on dividing the data set into subsets, then start to suffer, because if you want to divide a data set with a given number of images into a discrete number of subsets to describe a continuum, you'd need many, many different states to describe the continuum very precisely. And then the number of particles contributing to each of those states would become very small. So new methods that can deal with reconstructions that can actually describe the continuum conformational states, if you like, are now hotly sought after. And for example, Michael Unser here at Lausanne is one group who works hard on that problem. Okay, just to show you another thing, because we are doing this over Zoom today and not in person because of this virus, SARS-CoV-2, which causes COVID-19. And doing these type of reconstructions, being able to classify out different states and so on. For example, together with John Drex here in the lab, we looked at virus particles from infected cells, and in particular at the spike protein that sits at the top of this virus particle. So these are about 80 or 90 nanometers across, and then there's these spike proteins. These are the ones that form the first interaction with the human cells upon infection and the target of all of the vaccines that are out there. So knowing what the different states of these spikes are on top of the virus surface can be very useful for vaccine development. And I guess it's fair to say that vaccine development of all the vaccines that's currently being used was heavily helped by all types of Cryo-EM structures of coronavirus proteins, in particular this spike. Good. That was my rather long introduction. So in 2020, we kind of made, so that was last year, and the paper was actually written down from my attic at home during lockdown. We made another step forward. So I mentioned to you the big step forward of the cameras and the image processing, but in 2020, Thermo Fisher kind of was ready with 3 of its new hardware, so microscopy hardware developments, technologies, that were all combined in a prototype microscope that was operated by Abhay Kotecha in Eindhoven at the factory where the cryosys are made. So this microscope has a cold field emission gun, a stable Selectrix energy filter, and the latest generation of detector called the Falcon 4. Now, Abhay collected images on some test samples, and these were processed in my group by Takanori Nakane, who's a very talented postdoc. Now, before I tell you about the cold fag, I have to tell you that the objective lens in electron microscopes is not very good in terms of chromatic aberration. So chromatic aberration, as you may know, is the property of a lens that different wavelengths of the incoming light are focused in different planes. And here's just an example from optical microscopy, but the same happens in an electron microscope. And that means if the incoming electron beam that we use to image our samples with contains multiple different wavelengths, or it has a certain energy spread, then you get different defoci for the different energies in the image, and that leads to a fall of information at high spatial frequencies, just as you can already kind of appreciate from this example image here. Now, in the traditional cryos, now comes with an XFAC field emission gun. This is kind of the bit of the microscope where the electrons get generated for the imaging, and it has an energy spread which is given here by the blue curve, which has about 0.7 electron volts kind of full width at half max. By now operating the field emission gun at much lower temperatures, these are operated at high temperatures where the cold fag is operated more or less at room temperature, you can reduce the energy spread of the electron beam to only 0.3 electron volts. And that then has the effect that there's much less problems with the chromatic aberration, and the information content in the images gets improved, especially at the higher spatial frequencies. So that's shown here in blue for the XFAC and orange for the cold fag. So whereas if you use a traditional XFAC, after 2 angstroms resolution, the chromatic aberration of the objective lens start to become a very severe limitation for your imaging. Whereas for the cold fag, all the way up to 1 angstrom resolution, this is not much of a problem. The second new element was the stable energy filter here. Now, the image I showed has kind of a single energy of electrons coming in, which I just told you wasn't exactly true, but let's say that is. Then still, the sample, there's interactions of the electrons with the sample, and only 1 out of 3 electrons or so is scattered elastically and will remain the same energy. And those are the images that are useful for the type of images, the electrons that are useful for the type of images we collect. Now, the rest of the electrons, they scatter inelastically, meaning that they lose part of the energy in the process. They deposit that energy in the sample, leading to the radiation damage that was the problem of our images in the first place, if you remember. But so now they've lost energy, and now all they do is contribute noise to the images. So that's kind of indicated with yellow and red here, these are electrons that have deposited energy in the sample and are now contributing noise to the image. Now, by having an optical system here, which we call an energy filter, which is kind of in a prism based, you kind of bend them around the corner, and the ones that have lost energy are bent differently than the ones that haven't. So you can then put a physical slit here and filter out only the electrons that have not lost energy. And this technology has existed in the field for many years, but these filters weren't very stable. And this one developed now is much more stable than the ones that were available. It kind of only moves by about 1 or 2 electron volts over many days. And that means we can make that slit a lot more narrow than the slit in previously existing filters, which leads to the removal of more noise from the images. That together with the latest camera, which was even faster than the one before, I think the main advantage here is throughput, then allowed us to take images of a sample. This is a test sample. It's called apoferritin. It's a kind of cage-like structure that you have in your cells as well. These ones are mouse apoferritin, and it's used to transport iron around. So we took images, we collected 3,370 movies in about a day and a half, which is done much faster now, but altogether that gave 363,000 of these individual apoferritin images, which were then combined by Takanori into a reconstruction to a resolution of 1.2 angstroms, 1.22. And at that resolution, because the distance between many of the atoms in proteins, almost all of them, is more than 1.22 angstroms. That means that each atom is now represented by an individual blob of density, and this is what we then call a true atomic resolution. I have to say that in order to get here, you have to do everything right, and it was particular developments by Jasenko Zivanov, who's now in the lab of Henning Stahlberg, to track motion of the particles as the electron beams hit sample, which I mentioned about before, but also corrections for optical aberrations, which I'll mention again in a few minutes, that allowed to reconstruct apoferritin to this type of resolutions. Without corrections like that, we would have got stuck before reaching atomic resolution. Now, at these type of resolutions, you can even calculate difference map, where we have a model that does not have hydrogen atoms and a model, and then the map which does have hydrogen atoms. If you subtract the two, you can get a difference map, which then shows peaks for individual hydrogen atoms. We were very pleased to see this one, because this red atom is an oxygen, and these little gray ones here, they are hydrogens. So there is 2 peaks, 2 green peaks for the hydrogens and a blue peak for the oxygen. So 2 hydrogen and oxygen makes H2O. So this is an individual water molecule image inside the protein. You can see that the hydrogen peaks are not exactly where the atoms are, and that's because we're not actually looking at the atom itself. We're looking at scattering potential, and Gary Mershidov was very helpful in analyzing that actually you expect the peak to be not exactly where the center of the atom is. So that all made sense. We also used a new movie format, which allows you to collect these images at a frame rate of 248 frames per second, and we can now do reconstructions at a frame rate. So we can now look how you can do then reconstructions of individual frames of the camera, and you can see how the density evolves during a typical exposure of perhaps one or a few seconds, and thereby you can then start to follow the effects of radiation damage. For example, we know that aspartic groups get blown away. So they're present at very early stages of exposure, but then they get blown away from the protein due to radiation damage at the later stages. Another molecule we looked at together with the group of Radu Aricescu at the LMB was the GABA-A receptor, which we could do at 1.7 angstroms. And this receptor is very important for us all. It's in your brain, and it's related to your responses to alcohol, barbiturates, and all kinds of mind-altering drugs. So being able to study this receptor at this type of resolution, where you can really start to see all kinds of interactions at the atomic level between drugs bound in the pockets, for example, is very important. And even at this resolution, we think we can see hydrogen atoms in these difference maps. Cool. Before I wrap up, I thought I'd briefly mention also the other part of the group, which is work on amyloid structures, all very close collaboration with Michel Goddard in the neurobiology group. So this work was made possible, again, started from image processing work done by a very talented student, Sjors H.W. in my group, who implemented helical reconstruction in Milan. And this story is mostly about the protein tau, which aggregation of the protein tau into filamentous structures characterizes a family of multiple neurodegenerative diseases. There are probably more than 20 of them, which are called tau proteinopathies, and Alzheimer's disease is the most famous and the most frequently occurring one. So what we did, we took the brain of a 74-year-old lady who died of Alzheimer's disease, and Ben Falcon in Michel's lab isolated tau filaments from the cortical regions of this brain, imaged them in the microscope, and then using Sjors software, we were able, these are images of these filaments, we were able to solve the atomic structures of these tau filaments in the brains of Alzheimer's disease patients, for the first time being able to build atomic models to find out which parts of the protein are ordered, etc. And they form these very long helical structures where there's beta sheets along, very long beta sheets along the helical axis, which is typical for amyloids. I will not go into the details of the structures, but another perhaps famous tauopathy is a chronic traumatic encephalopathy. If you expose yourself voluntarily or involuntarily to repetitive brain injury, for example, through boxing, but I think in Europe, the discussion is more about headers of football. But for example, involuntary could be through explosions in the army. This is a problem in soldiers in the US, for example. Then you get this kind of environmentally induced form of neurodegeneration, very devastating effects at a very young age. People die of this. And they also have tau filaments. And we also looked at these. So we looked at tau filaments extracted from the brains of 2 professional boxers and 1 professional American football players who had died of CTE. And here, again, this is nice how then image processing developments directly slid into our experimental work. It was really thanks to work by Jasenko Zivanov, and I mentioned him also for the atomic resolution work, who developed methods to detect and correct for optical aberrations that might have been present in the imaging setup that you hadn't detected, but thanks to Jasenko software, for example, this was a Cryos at the Diamond facility here in Oxfordshire, and using Jasenko software, we were able to detect 3-fold and 4-fold astigmatism in the microscope setup, which was later on corrected by the Thermo Fisher engineer by realignment of the column. But thanks to Jasenko, we didn't need to recollect our data with the corrected microscope setup. We could actually use his software to correct for these aberrations, leading to a resolution now of 2.3 angstroms for the best CTE filament. And then you can start to see individual water molecules, only the oxygen atom, alternative side chains, etc., which kind of leads to additional insight into these filaments. So surprisingly, this is a structure from the football player, and these are structures from the boxers. If you have the same disease, you always have the same type of filaments. But if you now compare this to Alzheimer's disease, we also looked at multiple Alzheimer's disease, then the same different patients with the same disease always have the same type of structures, which I showed you earlier, but they're different from the structures in CTE. And even though this is the exact same protein that adopts amyloid structures. And the same is true for different diseases as well, different tau proteinopathies. We looked at Pick's disease and at cortical basal degenerations, which are different types of tau proteinopathies, but it is the same protein adopting these filaments. And you can see that for each of the diseases, a different filament fold is observed. So we don't know yet why that is. We would very much like to find out. For that, it would be great if we could make these different types of filaments in the lab, have some sort of in vitro system that we can play with, that we can tinker with perturb and see what happens. That so far has not been successful, but hopefully what we will find out in the future is how the different types of filaments get made in the different diseases. So I'll wrap up at that point. So that brings me to my conclusions. I'm very excited. We can now do Cryo-EM imaging to atomic resolution. This new generation of electron microscopes, 2 of which I just heard will arrive within a matter of weeks at the EPFL. So that will be very exciting. You can do great imaging on those machines. I think, but this is already a bit older since the beginning of the 2010s, I think the separation of different states in the computer, so the option to reconstruct multiple different conformations from a single mixture data set has really made a big change and has created a lot of new opportunities for structural biology. I think there's still, speaking of where this field will go, there's still a lot of potential there to be able to describe these continuous conformational changes that I briefly mentioned before. And just examples that I've shown you today of what's now possible besides that atomic resolution reconstruction of that kind of test sample, the very stable sample. That one doesn't move a lot, which allows us to go very high resolution. But I'm very much excited that we can now look at atomic structures of these amyloid filaments that cause these, that are characterizing these different neurodegenerative diseases. But as I briefly mentioned, for example, structural biology with Cryo-EM is also playing a major role in getting over this coronavirus pandemic by helping to make vaccines and hopefully allowing us all to soon meet in person again. So that brings me to my acknowledgement slide. I mentioned Abhey and Takanori's work on the atomic resolution. There's lots of people in Radu Aricescu's group also worked on the GABA-A receptor, which are not on this slide. Shauda did the helical reconstruction. Anthony and Ben did the initial AD filaments, and Ben and Benjon did some of the other structures of the other traumatic that I mentioned. Jasenko is now in Lausanne and has been very helpful in both projects, really, with contributing new algorithms. We get our brains from Bernardino Gatti in Indianapolis in the US, without which this work on amyloids would be not possible. And that work is all together with Michel Goddard. So I thank you very much for your attention. I'm happy to take some questions.